This is part 2 of chapter 8, where we've been talking about inherited cancers, and we've been focused on tumor suppressor genes being mutated as the cause of these cancers. And I wanted to point out that there are really two different classes of tumor suppressor genes. So all the examples that we've looked at so far, the P53, retinoblastoma, APC, would be examples of what are called gatekeeper tumor suppressor genes, meaning they control access to the cell cycle, right? They're acting as a gate or a door or a red light, however you want to think of it, in terms of the ability of a cell to be able to progress through the cell cycle. But there is a second class of tumor suppressor genes called the caretaker tumor suppressor genes, which are all involved with taking care of DNA damage, repairing DNA damage. And so these can also, mutations in these caretaker tumor suppressor genes can also lead to cancer, and these kind of mutations can also be inherited. So I wanted to show you a few examples of these. So, essentially, in a nutshell, because these caretaker genes are repairing DNA damage, if you mutate these genes, you are no longer able to make those repairs, and you're just allowing DNA damage to accumulate. And this can basically indirectly now lead to cancer because some of that accumulating damage, right, these additional mutations that you're picking up may be in oncogenes, proto-oncogenes, or some of those other gatekeeper tumor suppressor genes. And those kind of mutations can more directly cause these cancers. So just to give you a couple of examples, first example is xeroderma pigmentosum, which is this, again, an inherited cancer. And these individuals are intensely sensitive to UV light. So even just a few minutes exposure to sunlight, and you get very severe sunburns and skin cancer developing. You can see the face of this poor guy or gal with xeroderma pigmentosum, just the severe sun damage. So if you have this syndrome, you have about 2,000 times increased risk of skin cancer. Some other cancers are also pretty significantly increased as well. So if you have xeroderma, you basically have to essentially wear a spacesuit and be completely protected from the sun in order to be able to go out, right? These special UV shielding suits to allow them to go outside. So it would obviously be a tough way to go through life for sure. But yeah, that increased skin cancer risk is just pretty staggering. So here you're looking at the cumulative percentage of individuals developing cancer. It's basically the age at which everyone in a particular population who will develop cancer has developed some cancer. And you can see in normal human population that that's really not reached until age 80 plus, right? Basically, everybody who's going to get cancer has usually gotten it by the time they're 80, whereas in the xeroderma community, that's bumped up to early 20s, let's say. So, huge increase in cancer risk. And this is thankfully a rare recessive disorder. But this is really inherited in classic kind of Mendelian fashion, right? So if you look at the pedigree here, you'd have to have parents that are carriers, so each having one recessive allele, and you would have to inherit that mutation from each parent. So if both parents are carriers, as in this case, basically about a quarter of the kids would inherit both mutations and have this disorder. So the defect here, the mutations are in genes that would be involved with repairing pyrimidine dimers. You remember we talked about exposure to the UV light can cause these covalent bonds to form between adjacent pyrimidine nucleotides, thymines in this case. These dimers now distort the DNA, and when DNA polymerase comes along to replicate, it will insert the incorrect bases and basically lead to some permanent mutations in that particular gene. But luckily, we have this excision repair process that can identify and remove pyrimidine dimers. And so in a nutshell, the way this works is you have this enzyme called an endonuclease that would come in, cut out the damage on the one strand of DNA where the dimer occurred, leaves you with a little gap that can now be filled in with by DNA polymerase, right, using the other strand as a template. And then DNA ligase would come in and seal off the nick, and problem solved. So this looks pretty simple, but I did want to point out there's actually a lot of different proteins and genes involved with this excision repair process. And I'm not going to hold you responsible for all these details, but I just wanted to point out there's actually 8 different genes that could be mutated that could cause xeroderma pigmentosum. So it's a little bit more complicated. Come back here. So you can see a lot of these are named XP for the xeroderma pigmentosum. The 8th gene, this XPV, is actually this DNA polymerase delta that can actually accurately replicate DNA across a thymine dimer. So sometimes, if the dimer isn't actually removed, this particular DNA polymerase could essentially correct the problem just by replicating correctly through it. But obviously, if this particular polymerase gene is mutated, that's not going to happen. So that can lead to additional increased mutations as well. And that process is called translesion synthesis, being able to synthesize new DNA across that particular lesion, that dimer. So the way they figured out there were so many different genes involved in this process was by doing this common genetic trick called complementation experiments or complementation analysis. And I don't know if this was covered in your genetics class or not, but you could do this kind of analysis by crossing organisms together, or in this case, they're just simply doing cell fusions. And basically, the idea would be to collect cells from different individuals with xeroderma pigmentosum, and then in the lab fuse those cells together and see whether or not the resulting fusion cell is sensitive to UV or not. And the basic idea is if the cells that you collected from these 2 different individuals had mutations in 2 different genes, as you see is the situation here on the right, right, 2 different genes involved in this same repair process. But once we fuse these 2 cells together, now the cell on the right had a mutated version of XPF, but a normal version of XPA, whereas the cell on the left had a mutated version of XPA, but a normal version of XPF. So when we fuse those 2 together, we have a normal copy of each gene, so that fusion cell now is going to be resistant to UV, whereas the initial cells were each sensitive. So we would say those 2 mutations, quote unquote, complement each other, just meaning they're in 2 different genes. Whereas if you collected from 2 individuals that had mutations in the same gene, right, both XPA mutants in this case, we fuse them together, and now this cell still doesn't have a good copy of XPA, so that excision repair process is still broken, and the cell is going to be sensitive to UV. So just by doing these kinds of complementation analyses, you could get a feel for, okay, well, how many different genes are we dealing with? And in this particular instance, they realized, oh, okay, we're dealing with at least 8 different genes here, all involved with this same individual process. Okay, second example is another type of hereditary colon cancer. Um, and this one is due to mutations in another repair pathway. This one's called the mismatch repair pathway, which, as the name implies, is repairing mismatched nucleotides in the DNA, which could happen due to an error during DNA replication. Right, DNA polymerase inserts the wrong nucleotide. Usually it's pretty good about proofreading itself, but sometimes it won't catch a mistake. So the idea would be just to identify this as a mismatch and remove um, the incorrect base and insert the correct one. So if we catch that before a second round of DNA replication, it's no problem. We fixed it. If we don't, if we leave that replication error in place, now when we go through another round of replication, we're going to end up with a DNA molecule that has a permanent mutation. So you can see we went from an AT pair here to a GC pair down here because of that C that was incorrectly paired with the adenine. So again, this looks pretty straightforward, but similar to what we saw with the pyrimidine repair process, there's actually a bunch of different genes involved, 8 different genes they've estimated, again, due to similar kind of complementation type analysis. Um, so basically, individuals with this particular inherited colon cancer inherited a mutation in one of these 8 genes, right? And then you're going to get a random mutation in the second copy, basically the same kind of 2-hit type analysis in order to get that particular cancer. Third example is one you may be familiar with, the BRCA1, BRCA2 mutations in breast cancer. So obviously breast cancer is a huge problem. 1 in 8 women in the U.S. will develop breast cancer in their lifetime. But interestingly, even though there's been a lot of attention paid to these BRCA mutations, only about 10% of breast cancer is thought to be due to hereditary mutations. But if you do inherit one of these mutations, it can increase your cancer risk by up to 80%. So you can see, yeah, if you are born with normal BRCA genes, right, about a 12% cancer risk or risk of developing breast cancer. If you inherited a BRCA1 mutation, that jumps up to as high as like 65%. So what do the BRCA proteins do? Well, they're involved with double-stranded DNA break repair. And one of the initial studies that showed that this was likely to be the case was just looking at, well, where do the BRCA proteins localize? And we say, oh, well, they localize 2 sites of double-stranded DNA damage. So in this case there, you're looking at BRCA1 protein localization on the right, labeled with red, and on the left they have this control protein, H2AX, labeled in green. This is a histone protein that's known to localize to double-stranded DNA breaks. And what they did in those experiments was they just ran a laser across these cells, and that high energy from the laser will break the DNA. And you can see the BRCA basically localizing exactly to the path where the laser crossed and where the DNA double-stranded breaks occurred. There does also seem to be a pretty strong environmental component, even with those with inherited BRCA mutations. So there's been some interesting data showing that individuals that inherited BRCA mutations who were born before 1940 have a lower breast cancer risk than people who inherited BRCA mutations that were born after 1940. So that's suggesting there's some environmental change pivoting around that particular date. And, there have been lots of candidate factors identified, but I don't think it's entirely clear yet what that factor or those factors might be. So just to summarize this recessive versus dominant cancer risk syndrome. So classic recessive cancer risk would be something like xeroderma pigmentosum. And when I say recessive, I mean it's being inherited in sort of a classic recessive fashion, right? It looks recessive in a family pedigree. And again, in this case, you would have to inherit a mutation from each parent. So each parent would have to be a carrier. And it's basically a 1 in 4 chance, right, that this couple's kids are, that each child is going to have XP in this case, versus the dominant cancer risk syndromes like retinoblastoma, where, as we discussed, it is inherited in a dominant fashion, even though genetically speaking, by definition, it's really still a recessive situation because both copies of the gene need to be mutated in order for that cancer to develop. But this was the classic 2-hit model, right? You inherit a mutation from one parent, and then you have to pick up the second mutation during your life. So while we're on this discussion of mutations, I want to bring up this distinction between loss of function mutations and gain of function mutations. So in all these different cancers we've talked about in this chapter, these tumor suppressor genes, these mutations are causing the loss of function of the protein encoded for by these genes. So they're basically breaking the gene, breaking the protein, right? And that makes sense. If the job of these tumor suppressor genes is to suppress cell division, suppress tumor growth, then if we lose that function, then we're increasing the potential for tumor growth. Whereas you can also have what are called gain of function mutations. So these would be mutations that create some new function, or maybe just give more of the same function, cause excessive function of a particular gene. And this is typically what you see happening with the proto-oncogenes that will convert them into oncogenes. So you have your proto-oncogene, gets this gain of function mutation. Now you're producing too much of this protein that's helping to promote cell division, and that can lead to cancer. So kind of flip side of the same coin, right? In order for cancer to develop, you would need loss of function in tumor suppressor genes and gain of function in proto-oncogenes. Now, lastly, I also just wanted to mention that all the examples we've looked at so far in this chapter would be considered, high-risk genes, right? So if you inherit one of these mutations, your risk of cancer goes up by a lot. But you can imagine there are other smaller risk genes that would be a lot more difficult to tease out from, background environmental effects. So there have been some attempts to do this, mostly unsuccessful for a number of reasons. But like, for example, what you see down here was data from a study looking at cancer rates in the U.S. comparing black versus white Americans. And you can see data for cancer at all sites versus specific types, prostate, lung, colon, breast. And, if you look at the all sites, you might say, oh, okay, well, black Americans seem to be at a higher risk than white Americans. So, obviously there are some genetic differences between black and white people. So maybe maybe that's contributing to this difference. But if you look at the data a little bit differently, basically group it based on socioeconomic background rather than just pooling it all together randomly, now we see that increased risk basically goes away, right? So here you're still comparing black versus white, but you're lumping together those that had, basically didn't finish high school versus those that finished high school, those that had some college, and those that graduated college. And now, if anything, right, we're seeing the trend completely flip. So this would argue that there's probably something environmental there, because as soon as we were looking at socioeconomic status, right, whether you're black or white, if you're, rich or poor, you're more likely to be exposed to, rich people, regardless of race, are going to be more likely to be exposed to the same environments, and same thing for those of lower socioeconomic status. So anyway, bottom line is, it's really hard to distinguish some of these smaller risk genes from environmental effects, and so we may never be able to identify some of these genes that are playing a role, at least not with our current technologies. All right, so what about genetic testing? There are lots of genetic tests available, more and more every day. How do you know if a cancer that runs in the family, let's say, is hereditary or just due to random chance? Because such a high proportion of the population will develop cancer at some point in their life. So a few things to look for. One is if you've got several family members all developing the same type of cancer with no obvious cause. Grandma had breast cancer, mom had breast cancer, daughter has breast cancer. That would be a tip-off. For sure, if you're having childhood cancers develop, leukemia is really the one sort of common childhood cancer that doesn't necessarily seem to have a big genetic component, but for some of these other cancers, these more solid cancers, if they're developing early in childhood, that may be a tip-off that there's some genetic component there. Or as we saw with like Lee-Fraumeni syndrome, if you have multiple family members all developing multiple cancers in their lifetime, that could be another indication of genetics at play. So as I said, there are lots of genetic tests available for most of the known cancer genes, and there's some obvious benefits of getting tested, right? You could end some uncertainty. Maybe you find out you didn't inherit the mutation, and you don't have to worry about it. You might be able to get improved medical care. Get screened more frequently. You could make some better reproductive choices yourself, right? If you know you have or don't carry a particular mutation, might affect your choices in terms of whether or not to have kids. But I do want to point out there's also some downsides or potential risks as well. So, in some cases, results could be ambiguous, might cause serious anxiety or stress, right? Are you psychologically prepared to deal with a positive test if you get one? Is it a case where there's something that could be done? If you know you have this mutation, is there anything you could do about it? If not, then maybe it's going to be less helpful. Could lead to some discrimination from either health insurance companies or potential employers, and it could cause a strain in a family, right? If you're blaming family members for giving you some particular mutation and potentially some particular cancer, that can obviously stress those familial bonds. So anyway, just be aware some of these potential risks and benefits of genetic testing for some of these different cancers. All right, Chapter 9 will focus